I think in general, you can't save a species without saving a space for them to live. So this is a complementary method to making sure that they have the right habitat protected long into the future.
It's sort of in the end of the order of what should be done. But if there is a really sincere wish that we should get back species that we have had here before and where the habitat or the substrate is gone, if we can ensure that the conditions for that species will be present in the future, that we can manage the land in that way, then we have the option to do that. I think particularly for wood-inhabiting fungi, to grow them artificially and then to inoculate appropriate wood so that they can re-establish in areas where the population is very low or where they are gone. But the priority and what to focus most on is actually to manage what we have in the best way. And reintroducing is when we have failed and still want to get species back.
This one is Fellinus viticola. It's not threatened and it's not one of our inoculated species, but this is a nice species that it kind of prefers old growth forests and slowly growing trees. We have a large project of inoculation or translocation of threatened species here in Finland. We have altogether 20 species or 19 species which we have inoculated. We have inoculated spruce logs, we have inoculated aspen logs, we have inoculated pine logs, and we have also inoculated one species growing on living trees like Haploporus odorus. And over here in Espoo, in Luukki area, this is one of the places where we have inoculated spruce species. We have 5 different forests in southern Finland where we have inoculated spruce species. We have naturally fallen trees like this one, and they are all kind of decay stage 1 or decay stage 2 trees. So they are pretty fresh trees because we have previous research that were done by Nerea Abrek, Otso Vaskanen. I was also involved. So we have that information that when you do the inoculations in pretty fresh trees, so then they are more successful because there is not so much competition inside the tree.
I mean, putting species here and there, as we know from other organismal groups, it may have very bad consequences for the local communities or for the local species where the species are reintroduced. So in our research we are actually tackling this study question.
Yeah, so why are we doing these introductions? I mean, as a measure of restoration, I think the main motivation here is that currently we have such a huge extinction depth.
Yeah.
So that in these forests we would like there to be some species present that would be here naturally, but are not because of the very intensive forest management activities in the past. So now even if we do kind of restoration actions that produce more dead wood, those species don't find here. And now with this active inoculation we can bring the species there, and then they will be able to spread again to those new resources that we are producing for them by forest restoration.
The reason that this complementary method might be needed is we already live in a very fragmented landscape, and a lot of fungi are really limited by how far they can disperse. So maybe we start protecting habitats, but there's not really any good way for the fungi to get there in the next few lifetimes. Our lifetime, the forest scale, maybe they would eventually show up, but this helps them get a jump start and start a more biodiverse habitat more quickly. So yeah, we're only taking logs to forests that are in some sort of protection, where we know that they won't be cut or managed or anything going into the future. And we're focusing on species which don't have the chance to come there by themselves. So right now we've put mycelium in there. The long-term goal is, of course, that they colonize the substrate and that they produce fruit bodies and that they're able to spread. So I think in most situations at least within the site, we hope that they can continue and self-sustain the population. And then as we start to protect more and more sites in these areas and get more land coverage and more connectivity, then they should be able to spread into those other ideal sites which are not so far. So we don't see this as something that will continually need to be done to keep boosting up the populations, but just as a starting place to let them then help themselves.
Yeah, like we don't want to engineer like new ecosystems, but what we want to do is to help the species that could potentially spread here naturally. We want to just speed up that process by helping them to do that, and that's why it's very important that the sources are some as nearby populations as possible, because we don't want to bring genetic material to Finland from Norway, for example, but we want to have the main principle is that we introduce this species here through inoculation because it could spread here, but it's very unlikely given the current fragmented nature of this our protected area network and network of natural forests. But as I said, it's not to create some kind of a new artificial engineered ecosystems.
I actually have one with me over here. So these kind of wooden dowels, we put on each tree actually two wooden dowels that have the mycelia of the threatened species, and we put it inside over there, and then we cover it with wax. When we did these holes and we put these dowels inside the trees, we also collected sawdust. So from that sawdust we do DNA sequencing, and then after 1 year we have made one hole next to the inoculation points. We also DNA sequence that, and also 2 years after the inoculations we also DNA sequence that data, and we want to see if the mycelia has been able to spread from the inoculation points. And then we can also look if some of the species that were there before the inoculations, if they have disappeared. So we can look at the kind of succession or the interactions with different species, how they behave after the inoculation. That is also important.
So why it's very interesting to look through DNA methods which species are present in this log before the inoculation is that it really allows us to look at the influence of species interactions. That, for example, how does the structure of the community, the resident community before the inoculation influence the inoculation success, whether the newly inoculated species will eventually form a fruit body or not, and also how does the inoculation itself potentially kind of modify the resident community.
Yeah.
And now this relates exactly to this question that are we, for example, kind of causing some kind of an adverse effect to the resident community by the inoculation or not?
Yeah, it's two-sided. It's for that, and then also if ever this reintroduction method becomes a method in conservation practices from a methodological point of view to refine how it should be done.
One of the concerns that come up with fungal reintroductions is competition with whatever's in the forest already, and having enough substrate for the fungi that you want to bring and the fungi that's already there. So that was one of the bigger inspirations for this project, is to address that issue. We're not inoculating the fungi into any logs in the forest where there are things already living, but we're taking these empty, fresh logs. So this has both the benefits of bringing more deadwood into the forest, where maybe the forest is newly protected and it seems like it has good potential, but if it came out of management sometime in the past few decades, maybe it doesn't have so much deadwood yet. So this way we're bringing in more deadwood, which already support the fungi we're wanting to introduce, and then it will also be more substrate for whatever fungi come to join these logs, later stage decomposers, other things that depend on these species, and less concern to maybe drive out any fungi that exist in a log there. You don't want to take wood anywhere too far from the starting site where it was cut, because of course no wood that you get from the forest is sterile. There are things living in there or dormant in there. So you do want to be careful. The things that we considered are, do they have, for example, any beetles or anything, if we don't want to take them very far. So all of the ones that we were taking into southern Sweden were cut down in southern Sweden, and then from Finland, same thing. Well, we did DNA test at the beginning, so we can see already what is in the log. Yeah, and not taking ones that are clearly full of any pest or any known fungal pathogen or anything like that. We check before we start the work. I have willow, aspen, pine, and spruce. So 4 different hosts, all of which have a couple different fungal species that grow on them. And you might also notice there's a lot of birch. So we're testing them on birch because, for example, in Finland, birch is really common in forestry thinnings. They get taken out a lot. Easy to find kind of spare birch wood that would more or less be waste or just some cheap byproduct. So we're testing all of the species on that. High hopes that some of them can grow also there. So then maybe you don't need to find the really specific trees that they're found on in nature, but just to get them started, you can actually put them on birch, and then take them out to the forest where there is the host that they desire. Of course, doing fungal reintroductions, monitoring is a huge part that you want to follow up and see if it was successful and how successful. So part of keeping them on the farm is so we can care for them, but also it's easier to monitor. You have everything in one site and to see. One of our species, of course we can't say 100% until I put it on a plate and do some tests that it is the right species, but this pattern that you see is very telling, because these patches of mycelium line up with the rows where we put the inoculum. So here's an inoculation point, and there's mycelium on the end of the log. So if you see that, good indicator that it has spread, and this is only within a couple months.
Hey, here it is really nice. Now we are talking. There is one log that is already producing Fomitopsis rosea that was inoculated 2 years ago. Here we have a very nice example of one wooden bowl or section of a cut tree where we inoculated this red-listed species Fomitopsis rosea. And it was inoculated 2 years ago. And now, 2 years after, it has already started to make fruit bodies on the cut surface of the tree. So here we have small fruit bodies. It's a very beautiful species. It has like a pink underside. It's related to Fomitopsis pinicola, which is a very common species, but this is a rare species and beautiful species also. But this is very nice. There is this. You can see it is pink. The underside is pink. So that's really nice.
Fungi offer us an amazing opportunity to understand these dynamics of species-rich systems in a way that it would not be possible with many other organisms groups. If we think about one log, we have hundreds of fungal species inhabiting one log, and we can modify those species communities by introducing a species or by even changing a bit the conditions of the logs, and we can repeat these experiments at very large scales because we have so many logs that we can replicate a lot over space. So this is one aspect that is very important for my research, and why I think that wood-inhabiting fungi have a lot to offer to understand fundamental community ecology in general.
The wax and the sawdust gone. So I thought it would be way worse out here, but it seems actually like it's mostly better. So there's a lot of considerations that go into site selection. So of course you need the long-term protection. That's number one. You also want to pick somewhere that's within the fungi's existing habitat range or like historical range. So it's not our goal to bring them to new places. So you need to have a good knowledge of the ecology of the fungi and the populations, how they used to be. So you kind of need this red list monitoring to know where they were, how the population has changed, and then use that to select a site that makes sense where they really should be there. And then beyond that, you want different age structure, so that you know there's a continuity of substrate. So it's not enough to just have a lot of young aspen, for example, if you're doing an aspen species, but you want young, old, dead, young, freshly dead, old, dead, and you want this to be able to continue so that when they do fruit, they have, when they use up their logs, they have somewhere to jump to next. The species that we've chose do have a pretty well-known history. At least they've been studied in their ecology and habitats for a while in Finland and Sweden. And a lot of them do have trends on what kind of substrates they're found and what kind of microclimates they're found, stuff like that. So some of our species are almost only found on Kello, like these old barkless pine trees that took maybe 100 years to die before they ever fell down. It's a very unique substrate, and sometimes you only find the species on that in nature, but it's could be a question of opportunity, not requirement. So that's one thing we're studying. Obviously, we don't want to cut down a bunch of Kello trees for fungal reintroduction. We want to leave those where they are. So we want to see, can we bring them to the habitats on different substrates? So it's pretty common in edible mushroom growing that you don't have to grow them on the ideal host. So of course these are maybe easier fungi, but say shiitake, if you find it on oak, but we know it grows just as well on birch. But that hasn't been looked at with some of these species that are more rare, are more difficult, maybe more challenging, and they might really have higher demands than these edible fungi. So that's what we're testing. Can they grow just as well on normal pine instead of Kello? Can they grow even on birch, which in some areas is easier to get than pine? And then beyond just the wood type, there's a lot of other questions we're looking at. So some are found on really big logs, and the fruit bodies only come out in really late stages, which historically maybe some researchers had the idea that they only live in really old, but that might not always be the case. They might exist in really young or really fresh and just not fruit until they really need to once the log is quite decayed. So it's all these questions of opportunity versus need. So these are fresh logs, decay stage 1, if even. They are non-decayed at all. They were cut down just a few months before inoculation. And so we'll answer the question, do the fungi need the late stage to even survive? Do they need some of these fungi to like break it down and kind of transform the wood and create different chemical environments inside there before they can grow? Or can they grow okay and they just compete better on these more decayed logs? This is an aspen log, Radiolodon ericksoni, and same as what we saw on the farm. You see the same pattern. So really beautiful mycelium patches there, pretty clearly indicating that something's working in this log. Go do some tests and see more, but it's looking good. And it's also nice to see the comparison between like lab conditions, because that's another big part of my study. A lot of people haven't grown these species before. I would say most of the species, no one has grown, maybe on Petri plates, but not really the later stages. So a big part of the study is seeing how to grow them best in the lab, which will be really important for producing spawn to then transplant. It's not really something that you want to outsource, or that you can outsource to commercial growers, so you kind of need to develop those methods in-house. So it's cool to see that the ones that grow fast in the lab kind of like grow fast out here, and there are these like characteristics that correspond to each other. This is a nice old growth, lots of big old aspen here, lots of aspen deadwood. It's a quite rare habitat now in Sweden. But this is, one of our sites where we're introducing our logs. So these were inoculated the same time as those on the farm, and immediately after inoculation brought here. So all around the site we just have a lot of our little logs here with a tag that we can check all of the data, and happily growing fungi inside. At least some of them I see. We're testing like different ground contact amount, so some are totally flat on the ground, some are elevated a little bit because the fruit bodies often form on the bottom, but we're not stacking them. The stack on the farm is quite convenient for space and organization and stuff like that, but you get even quite different microclimate conditions from the top of the stack to the bottom of the stack, and most of these will assume that they probably like this close to the ground, more humid, kind of more natural contact with soil and other organisms, and higher humidity, and but we do still have the farm to compare, so we'll see. But for this farm, we're just going for for this site, we're going for a more natural kind of feel, mimicking the deadwood that you would find here. Another big concern is genetic diversity. Again, that applies to reintroductions of plants, animals, fungi, and with all conservation, I think genetic conservation is really critical, because you want populations to be able to adapt, because maybe this habitat that seems so suitable now won't be suitable in 10 years. Yes, it's really important to not just put out a single individual of a fungi, but it's important to have multiple strains, as we say, so different genetic diversity. So for example, in this project we took multiple, up to 5 strains of every species, and those are all in the same 4 sites together, so that they have the chance to breed, reproduce sexually, and increase genetic diversity. So we're saying, of course, you have to be careful, where do you take it from, where do you get the source populations. The guidelines are that you don't want to take them from too far away, but where is the right cutoff is open for discussion. What really matters kind of is how genetically different are the populations. So it might not be a matter of just physical distance, in case they're very similar over a large distance. And then, being careful to avoid commercial strains that you don't want to bring out. So a lot of the species we're working with have no commercial interest, have probably never been cultivated by anyone. And they also, because they're so weak competitors and they're naturally rare, so they're also threatened, they need help, but they're never common. They have very low risk of becoming invasive. The fungal geneticist that we were consulting with knew our species pretty well, and said that they don't vary enough between the Nordic countries to probably be a big risk. Of course, the ones that are in Canada vary a lot more, so the spatial scale is also always context dependent. Well, it would be really easy to take the viewpoint that we have no idea what's going to work where in the next few decades. So from that point of view, it's a little bit easier to some degree, let things evolve the way that they can, like let adaptation and change happen. And a rich genetic diversity, even very different genes, might help that, or it might hurt that. And so I think people have really strong opinions about this, but we don't have a lot of information. This is a moderate-sized research project, trying to take all of these different things into consideration. So we're also testing a north to south gradient to try to get some information on the impact of climate change, because we know that North Finland, North Sweden will become more like the South. So we're already testing, do some strains work better? Do some adapt better? Can they survive hotter, colder? Yeah, so definitely a research at this stage. Hopefully, like, it's a research and also big enough to have conservation impact at the same time, with also the chance to answer a lot of these questions.